The overall goal of the following experiment is to investigate oligomerization propensities of protein transmembrane domains, or TMDs, in a natural membrane environment. This is achieved by expressing the transmembrane domain of interest in E. coli as a chimeric protein with maltose binding protein for localization to the periplasm and ToxR as a transcriptional reporter. Transmembrane domain-induced oligomerization results in dimerization of ToxR and activation of LAC-Z transcription. As a second step, cells are lysed and treated with ONPG, which is hydrolyzed by beta-galactosidase to the light-absorbing species O-nitrophenolate, or ONP. Next, ONP levels are quantified by measuring absorbance of light at 405 nanometers in order to determine the levels of transmembrane domain oligomerization. Results reveal the oligomerization propensity of the transmembrane domains based on a ToxR reporter assay.
The main advantage of this technique over existing biophysical methods such as SDS page or fluorescence is that the behavior of the transmembrane domain is studied in a natural phospholipid bilayer by expression in E. coli, rather than a membrane mimetic system such as a detergent micelle.
This method can help answer key questions in the membrane protein field, such as elucidation of key structural features involved in transmembrane helix association.
Prior to the start of this protocol, pTox7 plasmids expressing the required chimeric protein are prepared from commercially synthesized oligonucleotides representing the TMDs of interest flanked by NHE1 and BAMH1 restriction sites. After plasmids have been prepared, gently thaw FHK12 competent cells on ice. Once thawed, transfer the cells into 15 milliliter culture tubes. Add 200 nanograms of each plasmid DNA to a separate tube and incubate the cells on ice for 30 minutes. Then,